starts with 10 warm-ups. We'll be holding our breath for a count of 8, and when we do positions, we'll do 3 reps on each side. Let's begin.
Are you wanting to lose weight, but nothing's worked? Are you starting to lose hope instead? Well, young Katie, the answer lies in epigenetics.
What's that?
What is the epigenome? Let's start with one very important example. Every cell in the human body contains the entire human genome, but a neuron is very different than an epithelial cell, and a lung cell is very different than a white blood cell. How do these very different cells come from the same genes? This is where epigenetics come into play. The epigenome is the collection of chemical compounds and proteins that direct the genome, turning genes on and off or controlling the production of certain proteins, while epigenetics includes all changes in gene expression without the change in the DNA itself. Some forms of epigenetic modification are temporary and reversible, while others are more permanent and can be passed on to the offspring. The cellular differentiation mentioned earlier is only one form of epigenetic modification. Epigenetics also encompasses methylation, phosphorylation, and histone acetylation, as well as interactions between DNA and miRNA, RNA transcripts, prions, and DNA repairing proteins. Most scientific research is done on DNA methylation and histone acetylation. DNA methylation is the attachment of a methyl group to carbon 5 of a cytosine ring in DNA, which is performed by an enzyme called DNA methyltransferase, or DMNT. Methylation most often silences a gene or keeps the gene from being transcribed or expressed because the methyl group prevents transcriptional proteins from binding to the DNA strand in the methylated region. Histone acetylation is the other major form of epigenetic modification. Histones are the proteins that DNA wraps around in the chromosome. They help the DNA from getting tangled and wind and unwind the DNA when it undergoes replication. When acetyl coenzyme A adds an acetyl group to a histone protein, the acetyl decreases the positive charge of the histone. Since DNA has a negative charge because of its phosphate groups, the DNA bonds are weakened and the DNA relaxes on the protein and become available for transcription. Histone deacetylation reverses the process by removing the acetyl group from the histone and therefore stopping transcription.
Okay, but what does that have to do with me?
As a matter of fact, young Katie, it may have to do more with your own mother, maybe even your grandparents.
Much evidence shows that many changes in the epigenome come from things parents or ancestors did before or shortly after giving birth to their children. Ancestral backgrounds can determine the chance of contracting a disease influenced by gene interactions, interactions between genes and nutrients, and the environment. A mother and father's diet, as well as any ancestors, can actually cause changes in gene expression due to epigenetic changes being passed on to the egg and sperm cells. For example, if someone's grandfather had an abundance of food in his earlier years, his grandchildren may be at risk for obesity and other diet-related diseases. On the other hand, if someone's grandfather had a shortage of food, his grandchildren may be at a lower risk for diet-related diseases, leading to healthier children due to the epigenetic changes of the grandfather during his time. An example of epigenetics where the offspring are already born is in bees. Larva that is fed a diet of royal jelly turns the DNMT3 gene off, allowing for queen gene to activate, while all other bees not fed royal jelly become workers and drones, even though both types of bees are born genetically identical. Mice that are cared for attentively by their mothers in early age are more likely to be relaxed in adulthood because methyl molecules silence the GR receptor gene in brain cells through methylation. Activities such as grooming can do this by the offspring releasing brain chemicals that activate cell signaling pathways, reaching all the way down to DNA.
There's no way this is all true. This sounds like some kind of backwards 19th century science. What are the proof?
Well, young Katie, I'm glad you asked.
Many people have doubted the legitimacy of epigenetics, stating that it seems very similar to Jean-Baptiste Lamarck's theory of evolution. Lamarck hypothesized that if an organism changes during its life in order to be better suited for its environment, then these changes will be passed down to its offspring. The classic example of this is the length of the giraffe's neck. Since giraffes feed on the leaves of trees, Lamarck believed that they stretched their necks until they could reach them, and this stretching was then passed on to the giraffe's offspring. Epigenetics is similar to this in that changes postulated post-birth are passed on to offspring. Lamarck's theory was disproved by Charles Darwin in 1859, so many discredit epigenetics. While epigenetic change can be passed down from parent to offspring, in many instances it occurs due to an individual's experiences. For example, a relaxed mother rat nurtures her pups more, so the pups grow up to be relaxed. The nature of the pup was dependent on the method in which it was raised, but not the mother itself. In most mice, the agouti gene is methylated, which produces normal, healthy, thin mice. However, when this gene is completely unmethylated, the offspring are obese, yellow, and more prone to developing cancer and diabetes. The two very different phenotypes of mice are genetically identical, only differing in the level of methylation of this one gene. One study on this showed how much the diet of a mother can impact her offspring. A pregnant yellow mouse was fed a methyl-rich diet, and her resulting offspring were brown and healthy. This research could be evidence that there may be more natural treatments for cancer, or that cancer can be prevented from developing in the first place simply by eating a certain diet. Polyphenols are found in certain foods, such as green tea, broccoli, and soybeans, and demonstrate regulatory activities in cancer cells. Research on human subjects is very difficult as each individual's genome is very complex, unique, and not well understood. However, there has been some progress.
You really need to warn me before you do that whole flashback thing.
Now that you know a little more, are you ready to make a change?
Well, it depends. What do I have to do? Whoa, how did you do that?
That is of little relevance. I believe it is time you learned a little more about exercise.
Research has shown that exercise affects the epigenome in many ways. For example, exercise can lead to changes in cytosolic messengers, which can activate signals to have changes in gene transcription. Physical exercise can also change the expression of different types of microRNA. MicroRNA are tissue-specific, non-coding RNA molecules which silence regular RNA and function to regulate post-transcriptional gene expression by silencing protein transcription and decreasing gene transcription. A recent study in Sweden found that physical exercise can also affect epigenetics by causing new methylation patterns. The experiment was set up where 23 individuals exercised one of their legs, but not the other for a period of 3 months. It was set up this way so each individual became their own control group, and other factors influencing epigenetics within one individual would not interfere with their research. Muscle biopsies were taken on each leg both before and after the experiment to determine what changes occurred in response to the exercise. They discovered new methylation patterns on the exercise legs, and that the majority of the new methylation patterns they saw were on enhancers, which amplify the expression of proteins by genes. The genes involved in the new methylation patterns were known to play a role in metabolism, insulin production, and inflammation of the muscles. What all this means is that exercise can reduce your risk of diabetes and increase other metabolic activities. There is also evidence that exercise can be a protective factor against cancer.
After all,
Keep in mind, young Katie, that you are what you eat.
I'm not gonna turn it into a fry.
Let's take a closer look, shall we?
Though it may seem far-fetched, what you ingest may alter your DNA. The correlation between genes and nutrition, especially in the realm of diseases and conditions, is known as nutrigenomics. Because everyone has a different genetic composition, food and drugs have varying effects on each person. Similar to pharmacogenetics, nutrigenomics focuses on how nutrients can alter DNA structure, sequence, and metabolism, which can in turn affect how a certain type of food is stored and excreted in the body. Alternatively, dietary chemicals can influence gene expression, with certain chemicals capable of creating new metabolic pathways and altering signal transduction pathways. More research must be done on this topic, since yeast has been the only organism studied with a recorded change in pathways of gene expression. Remember how we said earlier that a mother's diet can affect her offspring? In the U.S., folate is added to most foods to reduce the risk of neural tube defects in children. Folate is important in metabolic pathways that regulate gene expression through chromatin remodeling and methylation. If a mother fails to properly fortify her diet with folic acid, her children may contract spina bifida or another neural tube defect. While we're on the subject of diet-related diseases, you may be thinking of type 2 diabetes, in which a person's genetic coding prevents a proper synthesis of insulin to efficiently break down glucose into energy, or obesity, in which increased risk is associated with the FTO gene. Nutrition can also affect susceptibility to these conditions. Vitamin B12, folate, and methionine methylate DNA, decreasing the chance of insulin resistance, in which cells don't efficiently utilize insulin. Choline and betaine, which also influence DNA methylation, isolate histones of DNA. This leads to reduced risk of liver steatosis, which is the infiltration of liver cells with fat. Foods that are high in antioxidants can help combat free radicals, which are byproducts of biomedical processes of the body that cause damage to cells. Oxidative stress is an imbalance between the production of free radicals and the body's ability to neutralize their harmful effects with antioxidants.
So an apple a day really does keep the doctor away. Who knew?
Well, not quite. There's more to it than just eating right.
There's more?
Yep, some conditions can't be fixed with simple diet and exercise.
What could happen to me then?
Here, we'll show you. Thanks to errors that occur in DNA sequence, variations exist in genes. Epistasis, in which one gene masks or modifies the effects of another gene that controls the same phenotype, can play a role in disease risk. For example, if one gene metabolizes using a different pathway, this can affect other genes, increasing the chance of contracting certain diseases. Cancer is a major study within epigenetics because of the hope to reverse the epigenetic changes that cause the abnormal cell growth. Hypermethylation of the DNA leads to chromosome instability, tumor suppression genes being silenced, and chromatin being modified, which change the expression of the genes and invites cancer cells to the party. These changes in the genes from the environment can account for the changes seen that can't be explained by plain old genetics. Many of the epigenetic mechanisms previously mentioned, such as methylation, histone modification, microRNA changes, and phosphorylation, make DNA more susceptible to environmental changes. Due to this sensitivity, anything can trigger changes in expression, especially risks related to cardiovascular disease, such as smoking, lack of exercise, diet, and even stress, causing people with the factors to contract diseases more often than those who don't. Autistic individuals most notably have a decreased interconnectivity in their frontal cortex of the brain, but research has found that there are many epigenetic mechanisms that occur that cause autism and autism spectrum disorders. One main mechanism starts with the phosphorylation of histone H3 by DNA methyltransferases, which prevents demethylation of that histone to occur, creating a change in the expression of the genes of the histone. This process leads to a reduction of a gene's site's ability to condense chromatin and silence genes in the frontal cortex, circling back to the decreased interconnectivity.
No, but anyways, so I was in first grade, it was before class, and the straw had this bag of like craft rhinestones. And so we were putting them in our ears, and we were pretending they were earrings. We went to class, and I got one of them out, and I couldn't get the other out. 8 years later, I go to the doctor, and he's looking in my ears.
That whole time?
It was there that whole time. He's looking in my ears, and he's like, there is something lodged in your ear canal. So I had to go all the way to the ENT, like half an hour away.
Oh my God.
And I had to schedule an appointment, and she had this like huge microscope, and she had these tweezers. These tweezers. Oh my goodness, okay. What is that? What are you doing?
It's this new health shake. I read in a magazine that if you drink them every 30 minutes, it can help you lose weight. And I'm trying to lose a couple pounds, you know, for Halloween.
That's bull.
Sugar.